To our 24th webinar of bioelectrodynamics. And I would like to welcome everyone and announce that this webinar is also supported by the International Union of Radioscience, URSI, and also by Engineering in Medicine and Biology chapter of Czechoslovakia section of IEEE. It is my pleasure to introduce our today's speaker, Reuven, who is a professor at the University of Victoria. He is fellow of IEEE, Optica, and SPIE, and is also serves as a deputy editor for Optic Express and as a scientific advisor for ILF. That stands for
ILF. It's innovative light on fiber, or yeah, sorry, it's I'm blanking on the I. It's early in the morning.
Okay.
They're a company that does biosensors.
Okay, cool.
Yeah.
Cool, great. Reuven also served as a chair and co-chair of SPAN, Science and Engineering Symposium, and also as a chair of other conferences. But without further ado, I would like to give word to you. Please go ahead.
Thank you very much. Okay, well, thank you everyone for joining, and to Mikhail for having me at this seminar. I've seen some of the earlier editions, and they've all been really high quality talks, so hopefully I can live up to the standard. So I'm going to talk about a technique that we have for looking at single proteins, and in particular I'm going to spend a little bit more time on this ear technique, which we haven't really used that much in recent years, but because it is of particular interest to the dynamics of proteins, I thought I'd just spend a little bit more time talking about that later on in the talk. But I also want to focus on what other groups are doing as well, because there's been some great work by many other groups that have started to adopt the technique and use it and show some really interesting things. So I think it would be a shame to not present that. So I'm making sure to include some of that work. We did have the Near Field Optics Conference in Victoria this year. This is a picture of the attendees at that conference. There were 216 people in Victoria, over 100 from Europe. So it was great to have it, even with COVID still kind of looming upon us. We had pretty good turnout, and it's the 30th year that was held. So we're honored to be able to host that year. Okay, so let's see if I can advance slides. So I guess I probably don't need to motivate very much what we're doing here. But one of the key motivations for our work is to look at single molecule without labels or without modifying them at all, in fact. And the reason we don't want to modify biomolecules is because it changes their biophysical properties. So here is a really great work where they looked at labeled DNA, and they looked at some of the kinetics, diffusion properties, and the surface potential, and the impact of labeling, and they found that it actually changes all of these properties. So when you label something, you're not really looking at the thing that you started out looking at. So we want to get away from that. That being said, obviously a lot has been learned from works where proteins are modified to study their behavior. For example, in this work, they've tethered a protein onto a surface, and they've added two fluorophores, and they use FRET to kind of tell whether the protein is in the open or closed state, and they can see, they can monitor the folding and unfolding. But it's a really challenging experiment because you have to do all the labeling and immobilize the protein, but then you end up doing photon counting, and the whole point here is to just try to see how much signal you can get before you bleach or destroy the emitters here. So it is a very challenging experiment to do well. So what we're using is optical tweezers, and optical tweezers won the Nobel Prize, or half of the Nobel Prize for physics. I think it was 2018, and mainly because of their application to biological systems. And what they've been really good at doing is monitoring the forces and the dynamics of proteins and protein complexes in biological systems. So here are some examples with RNA, DNA, polymerase action, and kinesin walking along the microtubule. So all of these types of experiments used optical tweezers, but you can notice that there's usually this giant bead, polystyrene bead, or some other type of bead that's used to tether onto your protein of interest or your protein complex. And so this kind of really hinders a lot of the dynamics and adds this extra giant thing to the system. So you might not want to be able to do that. Another challenge with using optical tweezers is that if you want to trap very small things, like pretend you just wanted to trap the protein by itself, well, you'd be limited by the fact that optical trapping becomes weaker as Rayleigh scattering. So as your particles get smaller and smaller, the forces acting on them, the gradient force goes as a third power of the intensity. So essentially your optical power goes down a lot. So for example, if you had a 100 nanometer particle and you could trap that with say 10 milliwatts of power, if you went down to 10 nanometers of particles, you would now require 1,000 times more power. So basically 10 watts of power to get the same sort of trapping efficiency. So that is obviously a challenge that was recognized in the early works by Ashkin as well. Probably more serious a problem is this idea that the proteins themselves are small compared to the diffraction limited spot size of a laser beam. So if you consider a typical diffraction limited focus spot of being about 500 nanometers across, and a protein, in this case it was hemoglobin, being just a couple of nanometers across, then you can see it's much smaller than the laser spot. And now if you want to trap two of these, you won't really be able to distinguish one from the other. So the real challenge here is to trap individual proteins. So how do we overcome this challenge? Well, we use a double nanohole in a metal film. So essentially we have basically an optical tweezer setup where you focus a laser beam, but we focus the laser beam onto a double nanohole that we make in a sort of a 70 nanometer or 100 nanometer thick gold film. And then we can see the transmission of the laser beam through this double nanohole, the aperture. When a particle gets trapped, then it actually changes the transmission, increases the transmission. And so we see a huge jump in the transmission. And then you also see an increase in the fluctuations. And this increase is coming from the actual motion of the particle in the trap. So in this case, this is for a 20 nanometer polystyrene particle. It's quite easy to run this experiment. I'm just going to go through a video here that one of my postdocs made, where we basically take a microslide and put a double-sided tape spacer and then put some protein solution in there. This protein is PR65, which we've been studying in collaboration with Vitsaki and Bahar and Huang Shuang groups. Yeah. So we load in this in an optical tweezer setup, and there's our gold film that we've placed on top. So we can see the apertures that we've made in this this film, and I can describe how we make them. It's a really easy approach. But we find one of them and we align the laser to it. And then we adjust the polarization, and then we just wait for trapping. And when you see trapping, there's a very large jump in the transmission that we can see here. That's a single protein being trapped. And also the signal on the camera also changes a lot, so we can tell that we've trapped something. Okay, so that's a very clear indication that we've trapped something. So we've been using this technique for quite some time. I actually learned how to do optical tweezers from Reuven Kidon's group when he was at ICFO. Now he's at ETH in Zurich. But I was on sabbatical there, and he was trapping plasmonic particles, and I was working on nanoholes, and so we decided to work together to try to trap with apertures. And so initially we started with circular apertures. These were good to trap down to 50 nanometers or so. And then later on, I changed the shape of the aperture to really be able to trap single proteins. And this was with my student Yunjie Pang, who is now a professor in China. He showed the trapping of bovine serum albumin. So you see the trapping signal that we observed. This is over 10 years ago now. You have a jump when you get the trap in, but also this protein undergoes some conformational changes, and you can see transitions between this open and closed state of the protein. And to prove that we are actually transitioning between those two states of the protein, that was our hypothesis. What we did is we modified the pH. We went to acidic conditions where it's naturally in the open state. And in that case, we did not see these transitions anymore. It just, the jump went directly to the higher level. So that confirmed that indeed we were actually, the protein was opening up when it was in the trap. Okay, so recently several other groups have started to adopt the technique and look at it for different proteins. Case Decker's group has looked at this bow tie apertures, and they've also combined them with nanopore work. And here they're trapping two different proteins. And one of the nice things is that you can hold on to the protein for a very long time. So you can study and analyze it for a very long time when you have it trapped here. I was also somewhat involved in this work by Michael Meyer's group. Chui Fan Ying was the lead on this paper. It's still in the archive here. But essentially, what his group was studying was a bunch of different protein systems using a microfluidic setup. And in this case, he's flowing in hemoglobin, and they trap hemoglobin, and then they flow in oxygenated solution, and this allows you to actually see the binding of the individual oxygens to this hemoglobin. Another study, they did 4 really amazing studies where you can actually observe different dynamic behavior of different protein systems in real time without any modifications. So in this case, they're looking at comodulin, and this goes between the apo and the holo forms, and it's modulated through calcium. And you can see that when you add the calcium, it's a lot more stable here, even when they elevate the temperature. And they've used a really nice trick here of increasing the laser intensity at the aperture, and that actually increases the local temperature. So if you go to very high laser intensities, you can elevate the temperature where the protein is, and you can see changes in its transitioning between different conformations here. They also looked at this adenylate kinase, looking at this AMP, ATP, ADP enzyme action, and looking at all the transitions here. And finally, they looked at citrate synthase, and really showing some interesting statistics on the kinetics that you can't really see with ensemble measurements. So that was really an interesting point about their study. So it's clear from this that you can observe transitions on single proteins, observe them for periods of the order of hours, and you can also get really good time resolution with that. So all of these are really nice features of this type of experiment. So I just focused on two other groups there, but I'm also going to talk about Alexandrakis's group, University of Texas. I think it's at Arlington over there. I'll talk a little bit about what he's been doing with the nanopores later on. But there's about 20 groups worldwide that have adopted shaped apertures for trapping of proteins, but also for nanoparticles in general, quantum emitters and other types of particles. I won't go into all the details of what all these groups are doing. Okay, so a basic application of this trap-in technique is to measure the mass of the protein when you trap it. And one way you can do that is from this signature of the fluctuations of the protein in the trap. The amplitude of these fluctuations is related to the size of the protein, the mass of the protein that you trap, at least for globular proteins that we've seen. And we see a nice linear relationship here with different protein sizes. And actually, there's an interesting data point here where this is a complex of a serpine and trypsin. And what the trypsin does is it cleaves off an amino acid branch. And so this complex should sit at this vertical dashed line, but rather the mass sits at a slightly lower mass. And that's because this trypsin has cleaved off this amino acid branch, and so it's this peptide, and so it's essentially sitting at a lower mass than it originally was. So you see that you have kind of exquisite resolution on the protein mass here. And this is from Michael Meyer's group showing sort of the same thing, but using a different metric here. Instead of using the amplitude of the fluctuations, they're using the stiffness of the trap, which is related to the time constant of these fluctuations. So what is the correlation time? And from this, they also get this nice calibration curve with various different proteins. Okay, so we can compare this with other single molecule label-free techniques that are really emerging, and I think they're going to be the future of biophysical studies, biochemical studies, because they allow for studying proteins in their natural state without any modifications. And one is this NeoTrap. Again, Case Decker's group has pioneered this, but it's being taken over by Sonia Schmidt, I believe. And here they're using a DNA origami, and they block up a pore, and they do an ionic flow through this nanopore. And based on changes in the current, they can size the protein as well. Obviously, iSCAT is a prominent technique that's being successfully commercialized by Philip Kukura. And here you can actually size proteins on a glass surface by looking at the reflection and the interference between the reflection and the reflection off of the glass surface itself. And that gives you this iSCAT pattern that you can use to size the protein. And then we have our technique, which I've already shown you over here. Okay, so putting these all together, what we notice is that we have much better resolution right now. Actually, some people are refining the iSCAT and the nanopore techniques and pushing them down to smaller and smaller sizes. I believe I saw work from Vazid Sandagar's group where they managed to get the iSCAT down to 10 kilodaltons. But essentially, we can get down to well below 10 kilodaltons. And this is relevant because among all the proteins that are in the human proteome, the average size is 50 kilodaltons, and the median size is closer to 30 kilodaltons. So you really don't want to be limited by 30 kilodalton resolution, or I shouldn't say resolution, limit of detection here. Another nice thing about the trapping technique is you can work with dirty solutions. So we worked with things like just diluted egg white. And what's nice about that is you can trap and then release the protein and then trap again. And through multiple events, you can get the mass composition of all the different proteins in that solution. So you don't have to actually purify your solution beforehand. The trapping is a natural way to select out individual proteins and purify them. We've done a lot of work on protein small molecule interactions. So here's a canonical example of biotin streptavidin. Biotin's a small molecule here that fits inside these full barrels of the streptavidin complex. And when we trap streptavidin, it actually has a kind of a floppy behavior, kind of telegraphs between different states here. But when you add the biotin, it kind of locks up this floppy behavior. Some people who do molecular dynamics simulations suggest that the biotin actually comes into the barrel and closes the door behind it, so it kind of locks the whole thing up, which they believe is responsible for how this very strong interaction between the biotin streptavidin complex. We've also looked at small molecules that are drugs. For example, here you have acetylsalicylic acid, aspirin binding to cyclooxygenase, which is a protein in the blood. And you can see the time constant here. This is an autocorrelation. When you have the cyclooxygenase by itself, it has a much longer time constant than when you add the ASA to it. Of course, there's also some small molecules that do not bind with high affinity, so they'll bind and dissociate. So in that case, we can actually observe changes in the transmission through the aperture level when the small molecule binds and dissociates to this protein. In this case, the protein is human serum albumin, which is the human analog of bovine serum albumin. And by sort of tracking the amount of time that the small molecule is on the protein and off the protein, we can build up the kinetics of this interaction. So here are two small molecule drugs that interact with HSA, and HSA is not the target. This is an off-target interaction for these particular compounds. One's tolbutamide and one's phenytoin. And so by tracking the on-off binding, we can get the statistics, we can get the affinity of this interaction. And we notice that the values that we see looking at a single solution at a single concentration on a single protein, we get values that are very consistent with the literature reported values for the affinities here for these two different small molecules interacting with the HSA. So that's a very promising result, because these affinities, you'd have to do sort of a saturation curve to get them in the usual way, and it takes a lot of solution, and it takes a lot of you basically have to run the full titration to find out where you have saturated the binding here. We have also been working with Laura Itaki and Yvette Bahar and Michael Ohlmeyer, looking at some of the small molecules that are being developed for interactions with this PR65. That's the protein that I showed you in the video earlier, and we'll return to that protein a little bit later. But essentially, it's a important PR65 is part of the PP2A complex, which is one of the most abundant proteins in the body, and it's responsible for phosphatase action. These tricyclic compounds bind to this PR65 and reactivate it. So they make this phosphatase, they reactivate the phosphatase action, which is relevant for cancer treatment, because one of the first things that cancer does is turn off the phosphatase action, which slows down metabolism. So, I should say what we're actually observing here in this experiment. Well, we first observed the trapping of this PR65, and then Samuel, the student who's working on this, introduces a small molecule into an open chamber, and we see a second jump when it actually binds to the PR65 here. And so we studied more of this interaction in different contexts. We've also looked at protein DNA interactions. So here's a single-strand DNA. When you trap a single-strand DNA, you get a jump like you do with proteins. This is 20 bases. But if you trap a hairpin of that single-strand DNA, you see a jump which is about half the height, and then you see a second jump up to the full height, and in the transmission through the aperture. And this second jump we attribute to the unzipping of this double-stranded or this hairpin DNA. So we see it going from a hairpin into a single strand. Now, this is interesting for studying interactions with DNA, because certain proteins will bind to the DNA. For example, P53 binds to the DNA. And when we add P53 to the mix, essentially we see this intermediate step, or this intermediate region, increase by 2 orders of magnitude, okay, up to 2 orders of magnitude here. And so what the P53 is doing is suppressing the unzipping of this DNA here. And then we look at mutant versions of this P53, excuse me, which are ineffective in suppressing the unzipping, essentially you see that the DNA unzips with the same time constant, essentially, as when you don't have the p53 there, if it's the mutant. And this is just a single point mutation. It's not significantly altering the protein, but it really does significantly alter its function. This is relevant for a lot of studies where people are trying to target this particular protein and sort of reactivate it by metallochaperones as drug candidates to try to fight cancers again. It's a natural anti-tumor protein, this one. Okay, we've also looked at antibody interactions. This is a simple experiment we did early on where we took bovine serum albumin and trapped it, and then we flowed in an antibody to bovine serum albumin, and we noticed a second jump over here. And we confirmed through non-specific experiments that this was indeed binding and not just co-trapping of two different moieties. Related to this antibody work, recently there's been some great work by Alex Andrakis' group, where he's kind of combined the double nanohole architecture with nanopores, and then you get both the nanopore signal and the optical trapping signal. And he's used this to study, again, immunotherapy targets for cancers, and a number of other things. He's published several works on this particular, using this particular platform. But what's really nice is you can distinguish between the antigen, the antibody, and the pair of the two that are bound together. So that's a really nice feature here. And you can compare as well the current signal you get from the nanopore and the signal you get from the optical transmission. And here he's mainly looking at step height, not looking at the things that we focus on, which are the fluctuations in the time scale. But looking at step height, he can see a clear distinction between the antigen and the antibody here in this curve over here in part B of this curve. Whereas if you look at the nanopore information, it's sort of the antigen and antibody data lie on top of each other. So there's not a very clear distinction that you do see with the optical tweeter technique. So that's kind of a nice feature of this double nanohole aperture. It allows you to actually see and distinguish what you're trapping here and what's going through the nanopore. Okay, so Michael asked me to kind of focus a little bit on extraordinary acoustic Raman. So I'll spend pretty much the rest of the talk discussing that technique and our latest results with that technique. Okay, so as you all know, small particles have acoustic vibrations, basically sound waves, and you can kind of estimate the frequency of these sound waves by taking the dimension and the speed of sound in that particle, and doing a quick calculation. So, for example, if you have a particle that's a nanometer across and the speed of sound is a kilometer per second, then you're gonna have a picosecond sort of time for the sound wave to travel across, and so you expect to have vibrational resonances in the terahertz regime. Okay, so here you see C60, and it has these L equals 2 and L equals 0. That's the azimuthal quantum number symmetry. They have 8 terahertz and 15 terahertz vibrational frequencies. So we perform a similar, well, the actual experimental technique is not similar, but the results we get are similar, where we do the same double nanohole trapping, but we use 2 different lasers, and the lasers have slightly different frequencies. The reason we have 2 different laser frequencies is because when you interfere them together, that introduces a beat frequency. And this beat frequency, it turns out, when it matches the vibrational resonance of the particle that you've trapped, you tend to excite the particle. And what we observe empirically is an increase in the fluctuations of the particle in the trap. So we increase in this noise amplitude, in other words. So that's kind of how we find the vibrational resonances of the nanoparticle. We scan one of the laser frequencies until we see an increase in this noise amplitude, and that's the signature for the vibrational resonance. Okay, so when we first tried this technique, this was developed in my group by Skylar Wheaton. We looked at things that we knew the vibrational resonance frequencies, what they were. For example, polystyrene spheres. We could use Lamb theory to predict what the frequencies would be, and then we could scan the beat frequency of the lasers and see these peaks at those values. And so what we have here are the experimental values and the theoretical values for those particular size standard polystyrene spheres. Now, one of the nice things about this, and perhaps surprising, is that the peaks are rather sharp, even though this is all happening in aqueous medium. Whereas if you take conventional Raman spectroscopy, you're typically limited by the resolution of your spectrometer. People have done Brion scattering on particles down to about 200 nanometers in size. Maybe people have pushed that even further now. But essentially, with Brion, you could get similar types of resolution, but nobody's really applied that to single proteins. So here we're getting really sharp spectra to get the vibrational resonances of these nanoparticles here. But then we went and applied this to different proteins, and these are just size standard globular proteins that we had. And you can see this experiment over here. The top two data sets were repeated 20 times. So you can see the reproducibility of the data on different trapping events. And you get these nice spectra, which are different for different proteins. So we've seen some interesting normal mode vibrations here that have been predicted a lot, but there's really not too many other ways of observing these in experiment. And so I had a student, Tim DeWolf, try to come up with a theory to explain these vibrational resonances. And he used an elastic network model, normal mode analysis, and then tried to calculate what would be the Raman activity of these of the vibrational modes in this using this elastic network model, and then compare that with what we actually saw in experiment. And these are the data that he got. Not bad correspondence, but there is a lot of tweaking and fitting that goes into these models. So this is far from perfect, and there's a lot more to be done in this area, I believe. But it was a really nice sort of first study on this. Probably more convincing than proteins is to look at, again, single-stranded DNA, because that's just a linear chain of amino acids that are essentially connected by springs, and the spring constant is well known from pulling experiments, AFM and such. So we did the same ear experiment. This is by Abe Kotnaler. He ran this experiment on single-stranded DNA. And what he observed was, again, when you scan the laser frequency and you hit the beat resonance that matches the vibrational resonance of this linear single-stranded DNA, you get this increase in the noise or the amplitude of the noise that we see that's indicative of going through this resonance. And one of the nice things about this is you can change the length of the single-stranded DNA just by adding more bases and compare that with theory. It's just a linear lattice vibration theory, 1D lattice vibration theory. And you get nice agreement in the experimental results with the expected values. But also, the individual bases actually have slightly different masses. And so you can get different resonances even for the same length of excuse me, the same length of single-stranded DNA. So that's if you now think about looking at the entire spectrum, not just the first resonance or the second resonance. In this case, we're tracking the first resonance. You can imagine that this could be a way to fully tell which bases are there. So to sequence that strand of DNA. We've also, this is Jeff Burkhartsmeyer. He joined my group for a bit. He is a visiting student from Hong Kong, and he trapped polystyrene spheres, but then also trapped this Phi-X174 viron, which is a bacteriophage. It's quite a small a 25 nanometer virus particle, and so he could size it from the vibrations or from the motion of the particle in the trap, but then he could also track and find the vibrational resonance at around 30 gigahertz here. And so this is really my last slide here, where this is our recent work. Again, I'm looking at this this PR65. This is done by Elham Babai in my group, and she repeated all the calibrations on the polystyrene spheres, and then went and applied this to this PR65 protein. And the interesting thing about this protein is it's kind of it's spring-like. It has these repeats, these heat repeats that basically give it a spring-like quality. And this is being investigated for its action as an enzyme. Essentially, this spring backbone, it recruits an enzyme or something that binds to the protein that's it's acting on, and as well as something that is responsible for the phosphatase action. And so it gives specificity for the phosphatase action in this whole PP2A complex. So this particular spring-like action of this protein has been implicated in this enzyme action. So its spring qualities are important, presumably important for how this functions, for the allostery of this interaction. And so here we looked at the vibrational resonances using ear, and the vertical lines are the expected frequencies that were calculated using a sort of a normal mode analysis technique from Yvette Bahar's group. It says University of Pittsburgh, but she's now moved to Stony Brook. But essentially, the agreement is not bad, but there are some deviations here, and we really have to work more on this data to get it really nice. It's very sensitive. The technique is very sensitive to the intensity that you use and the optical tweezer, the specific setup. So and the lasers, you got to be very careful that they don't have feedback into the laser. So there's a lot of things you have to really do carefully to really get nice data. And so this is one of the examples where I think we got really nice data, but we really have to do more work to sort of flesh this out. So that brings me to the end of my talk, basically 40 minutes there, I believe. So I'm happy to talk more about how we make these apertures or other things, but otherwise I'll just take some questions.
Okay.
Thank you very much for a nice talk, very interesting. So if there are, let's see if there are any questions from the attendees, or I'll start. I have several. Oh, there is a raised hand by Vladimir Beres, so Vladimir, I just I make you presenter. You can ask yourself if you like. Or Vladimir, you had a question? I see your had a raised hand, or just us by.
Okay, can I?
Yes,
yes.
Please.
So my question is, how small should be a molecule what is being attached or bind to a protein that you can still see in your images? Like, is there any kind of the bottom size or smallest mass of that small molecule?
Oh, okay, that's a great question. So and the short answer is I don't know. So usually when we bind these small molecules to proteins, what we're really observing are changes in the protein, not really the small molecule itself. So so I do not believe that we can directly relate the change in the signal we observe to the mass of the small molecule that's added, but rather it's more indicative of the properties of the protein, I believe. For example, going from a floppy sort of state to something that's a little more rigid. We have observed trapping of proteins down to 4 kilodaltons in size. So that's neuropeptide Y, and we've sized that. Now, neuropeptide Y is a very rod-like protein or peptide, and so so 4 kilodaltons is kind of the limit of trapping a single protein that we've achieved so far. Now, potentially we could go even smaller. We've discussed with collaborators looking at lipids even, but this is that's the size that we can size it nicely. It doesn't give it gives a much larger noise amplitude because of the rod-like shape, but the time constant is well correlated with the size.
Thanks. May I ask another question, just a very short one? Is there any difference in the sensitivity of your method depending on the type of overall structure of a protein? Say, if it's fibrillar, for example, or if the protein is made up of, I don't know, layers of beta sheets?
Yeah, so we have observed some differences. Like I just mentioned, neuropeptide Y is very rod-like. It has seems to have a larger amplitude of scattering. Now, you can really use this technique, I believe, to probe the hydrodynamic properties as well as the optical scattering properties. So you get both of those information. That information is contained within the scattering signal with the time constant and the amplitude. So the stiffness will give you information about, the stiffness and the drag on the protein will give you information about its both the optical scattering and the drag, so the Stokes drag. With globular proteins, they seem to size really nicely, but with this PR65, it seems to be slightly different. So it's a repeat protein. We've also trapped recently, and I didn't present any of this work, and it's kind of too early to present right now. But we've also been looking at G receptor proteins, so membrane proteins on these sort of HDL rafts. So encased in a raft of a membrane, and so we trap the entire thing, and we can observe that as well. So the signal will vary a little bit based on the type of protein they're using. When I'm talking about sizing, it's mainly looking at globular type of proteins.
I see, and in that concern, the very last question. You just mentioned that you tried to trap and track behavior of a membrane protein. It was a kind of membrane attached or so-called integral protein embedded into the lipid bilayer. I mean, is it possible to apply your technique to?
It was an integral protein. It was a G receptor protein.
So it's still there's hope that this will be possible to study conformational dynamics of membrane proteins.
Yes.
Thanks.
Actually, the tricyclic compound that we were studying for PR65 actually acts on those membrane proteins as well. So it's kind of an interesting side product.
Thank you very much.
Okay, so if I may, another question from my side. I'm wondering how is that these double holes in the in the metal. So it's like you have array, or how is it like? How many you have them on the let's say on the
Okay,
slide?
yeah. So let me switch to that part of the. I have some slides on that, I believe. So let's see here. So this is we fabricate them by this technique where we just deposit polystyrene spheres on a surface, and then we evaporate gold. And if you do it at the right density, you get a certain number of dimers.
Okay.
And we look for those dimers because they produce the double nanoholes. When you evaporate, it forms a template for the gold. So we just evaporate the gold over top, and then we sonicate or use the tape method to remove the polystyrene spheres afterwards. And this leaves behind these double nanoholes. We also have done plasma etching, oxygen plasma between the deposition, the evaporation stages, and that allows us to reduce the size of the polystyrene spheres and reduce the cusp separation as well. So we can finely tune the cusp separation down to about 10 nanometers. And so you can see the density here. You typically have, within a typical field of view of a microscope, you'll have about 6 double nanoholes there, or so, depending on the density that you fabricate this with. Here we did a constellation map. So what we did is we'd look in the microscope in panel B here, and we'd find a certain pattern of double nanoholes, and then we'd find the same pattern, or a certain pattern of holes, I should say, and we'd find the same pattern in the SEM, and then we would go and figure out which ones were doubles, which ones were triples, which ones were singles, and we studied them in that way. So you can see like a single here, or a double, some sort of more complicated structures. And by looking at the polarization of the laser through this aperture, you could really tell which ones are doubles. So it was a nice way to kind of to isolate the double nanoholes. And then it's also a really cheap way, because we used to use focus ion beam milling as a way to make these. This is a really cheap, fast way to make like hundreds of samples in a short amount of time.
Okay, that's cool. That's really nice. Yeah, it's only to some stochasticity, but we only have to define those which work, and that's fine. Okay.
Yeah, they
cool.
show up very clearly based on the intensity and the polarization.
Yeah, I see. Okay. My other question would be on basically inside what's happening in these double nanoholes. So it's really, I can imagine it really focuses down the field to the gap. So is that for your laser powers, like what are the field strengths you are getting there? So is it like for that, like it's like bigger question, like what's one thing like what's happening with the protein from terms of, let's say, about the thermal perspective, like is that like heating? Don't you observe like denaturation for certain proteins or certain powers, or along this way, like what's happening to the protein from a thermal perspective, also maybe from field strength perspective, it would be extremely high.
Yeah, that's a great question. So so the thermal part fortunately has been studied by 3 other groups in detail, 4 if you count Michael Meyer's group. So Case Decker, he combined, I mentioned this bow tie with a nanopore, and nanopore also gives you a measure of the temperature based on how it changes the current, the ionic current, right? So that was one way where they could track the temperature dependence based on laser power. Then Ken Crozier's group looked at some fluorescent particles and diffusion. And Wenger's group looked at also fluorescent particles, but more properties related to their emission. And using these different techniques, as well as COMSOL modeling of the heating, all of them kind of came to the same conclusion, even though they were using different systems, that you get basically something of the order of a degree Kelvin per milliwatt laser power. This is at slightly different laser wavelengths and all in the near infrared, but different laser wavelengths. And it is sensitive to what kind of adhesion layer you have, whether your laser is incident from the adhesion layer, which is typically not as good a metal as the gold, surface. So there are some differences there, but the order of magnitude is like a degree Kelvin per milliwatt. So to raise the temperature just to physiological conditions from ambient, you need 10 milliwatts of power already, and so or more than that. And so typically we're only getting to sort of ambient conditions. But I did mention that Michael Myers group, they really cranked up the power and they pushed it up above 60 degrees Celsius on purpose. And then you do see something happen, right? You see these changes in the protein. So yeah, that's a great way to probe that. And a lot of what people do with proteins is these melting type of experiments, right? You sort of measure the stability based on the temperature of melting and unfolding, or you add some other perturbation. So it's a nice way to do that. Now regarding the proteins. Thermal behavior, I mean, proteins are typically, they have interesting thermophoretic dependence. Some of them are thermophilic. It depends on the temperature as well. They transition from being thermophobic to thermophilic, actually, as you go, a lot of them, at least the literature that I've seen. So if you actually increase the power, you can actually have the situation where you enhance the ability to trap proteins because it pulls them into the trap somewhat. Now, about the actual laser, the near field power or the actual electromagnetic field that the protein's seeing, there is some impact of that. This shows up through electrostriction, right? So if you have a change in the polarizability, then if you're in a field, this will give you there's a force that acts on the particle to elongate it. And so we do tend to see, this tendency to unzip, for example, the double-stranded DNA, right? The hairpin, it unzipped. We do see these transitions of PR65. It sort of jumps. Bovine serum albumin tend to go into the open form, although it did go back. So the fact that it transitions and goes back kind of tells us that it's not a very strong force. It's not like an irreversible kind of force. It's somewhere at the kind of below the KT kind of limit, but better than that, we can't really quantify it.
I see. Okay, that's also related to the few times you mentioned the long-term observations, also which other group tended. So what's the limiting factors there? Like it's typically, well, you're having some would it be some photothermal damage, or what do you think is or just the trapping is not that long term, or
you can trap there is no limit. Like you can trap for days. The limiting thing that we have is just the drift in the piezo of the stage.
Okay.
So the student or postdoc has to sit there and kind of readjust it to pull it back to the maximum transmission. So that is kind of the challenge there. But if you had a very stable or you automated that process, like from a energetics point of view, it's going to stay there, and the laser intensity is not such that you damage the particles. So in principle, the observation time could be days or even longer.
Okay, yeah, that's really interesting.
Yeah,
I'm not sure I remember of other technique which would be providing much stability. So Volodymyr, you have a question, so go ahead.
If possible. So my question is how long it takes to, for example, pinpoint a single protein in a mixture. You mentioned once that you worked with egg white, for example. So my question is whether one day you can compete with, say, mass spectrometry for proteomics approaches for characterizing, say, mixtures and like performing the full characterization of certain protein mixture.
Yeah, so that's a great question. So I guess we wouldn't be competing with mass spectrometry because mass spectrometry itself has a lot of limitations. Actually, we have a really excellent researcher in that area at UVic, David Goodlet, who runs the proteomics center here, and they basically do mass spectrometry all night and all day, literally. you're limited in size. You need lots and lots of sample. People run like other types of methods like gels or whatever to figure out the protein compositions. So it's a great question. So essentially, if I were to summarize, how long does it take you to trap and release a protein? Like that's kind of really, and then trap another one, like to repeat that process. And it can take a long time. And sometimes it's very like trapping happens at the surface, so there's a lot going on there that we don't fully appreciate. There's sort of double layer type of effects. There's thermophoretic effects. There's charging, other types of charging effects. So we really kind of tried to take account of those things. And we've also recently, and there's a guy just this in the cafe who's done a lot of work with trying to exploit this, applying potentials and doing other things to make use of the thermal effects to try to manipulate things. And we've been working on that a little bit, and we believe we have a technique which hopefully will get published soon. It's in review right now, and I'll probably talk about this at the biophysical meeting on Saturday. But essentially, we have a technique where we can trap things, basically make it an order of magnitude faster. So we can reliably do it in a minute or so. And that requires, some sort of electrode-based type of method. Obviously, now Alexandrakis and Case Decker have gone and combined this with the nanopore. There's extra complexity in doing that, but then you're using the nanopore flow to essentially bring the protein to the trap, and then it's really fast, right? Presumably they have no trouble doing that. We don't have the expertise to do that sort of stuff in our group.
Okay, so any more questions from the audience? So
I'm surprised you're not asking any questions about the ear stuff, because that was the whole point, I thought.
Yeah, well, it's definitely some. I think people maybe are just shy to ask at this point, but yeah, definitely.
Oh, okay.
That's definitely was something was several different of interest for. So I'm wondering there. Maybe it's follow up what we discussed last time. I was checking the paper of yours on these damping.
Yeah.
So the idea that you could have such a sharp peaks would be consistent with idea that consistent with the possibility of that these modes are underdamped potentially, right? This is the
right.
So then it's something very, very interesting as we chatted last
right
last week.
Yeah.
And I'm wondering if you did show that if anybody ever did similar analysis like you, and you showed in the wetting of the nanoparticle and in that paper, if somebody did something similar on a proteins. Are you aware of anything of that?
Yeah, not that I know of. I think proteins, there's obviously a lot of effort placed on getting a good solvation interaction, and so that would be something great to study. But typically, people who do molecular dynamics simulations don't really drive the motion of the protein. They just kind of rely on natural motion.
Yeah, that's exactly the thing I'm addressing. Because something they are also up to, and there are some studies which actually do drive it, but usually maybe much lower times, because not really driving it oscillatory as well in a way. So it's I think it's could be really interesting to see that.
Yeah. I mean, there's a bit of a disconnect. I know that Sine gave a talk in one of these series, and he's obviously done a lot of work on this no mode stuff. And there's a bit of a disconnect in the quantitative values that he was predicting things at a much higher frequency. And so it's a question, what is the impact of the aqueous medium, which is usually not included in these normal mode type of calculations. So, And I think it can have a lot of different impacts. So one impact could be, if you do have damping, then you not only do you broaden out the peak, but you also shift it to a lower frequency. But then the question is, well, why do you see such sharp peaks in our experiments? And so
exactly.
There is this idea that there might be some de-wetting, and so you essentially have like almost free oscillation. So it is a kind of an open question, and we don't really understand that well. But the data is kind of the data, so we have to kind of work with what we can observe and try to come up with other ways of doing it, which we are working on right now.
Okay. So I understand it's like also from the video actually looks cool, like it looks like simple in a way. So you have just we also touched upon that last week too, that if you have like is there any preferred also the charge of the protein? It's like the surfaces you use are preferably it's easier to work with the negatively charged or positively charged proteins.
Yeah, so I don't think we've really paid much attention to that. Maybe we should. When we first started doing these experiments, we did kind of think, okay, well, how do we make sure we don't get sticking on the surface? And so we did like MPEG thiol type coatings. It's basically peg thiol. And we found that it didn't really make a big difference. And part of the reason is because usually the surface picks up the same charge, typically negatively charged, although there are proteins that have different charges, right? And so then you could think, well, maybe they just foul in the surface, right? But once they fouled the surface, then that kind of creates a protection layer, and then you can go and trap after that point. But if you did want to look at positively charged proteins, you probably have to, because the surface usually picks up a negative charge, at least the underlying glass layer does.
Okay.
Then you probably want to do some treatment to account for that. But we haven't paid much attention to that. I think most of the proteins we looked at have been negatively charged so far.
Okay, I see. So the metals yours is it gold, right? The one which forms the double on the hole, or is it gold?
Yeah, we typically use gold. It's very stable. And, yeah, people
so
use
there,
a lot.
I'm wondering if you is there any time to stabilize the experiment before you started in a way that if you just had solid proteins, they should start to get absorbed, depends of course on concentration. They could also form like a continuous layer if you have enough proteins there. So did you observe something like drifts, which are not due to the thermal stuff, but because of the proteins get absorbed, or did you care about it at all? I mean,
we haven't really seen anything like that for the laser powders that we were looking at. So
I mean, like it's just because of the interaction
over
with the surface,
time, yeah.
Even
So I shouldn't say that. Over a long time, you will see that, and we do see that, and we've actually published that egg white paper is a good example there, because NOAA was doing a lot of different trapping experiments there, and we showed, over a long time, if you put it back in the SEM, you get this, like you can actually see the filth or the protein layer kind of forming up. So yeah, there's a kind of a lifetime to these before you have to clean them off or do something with them. So we have thought about that, but typically we try to work with like kind of fresh new samples and trap to the experiment that we're trying to do.
Okay, yeah.
Mm-hmm.
Okay, cool. Yeah. So, are there any more questions right now from the audience? One more from Vladimir. Go ahead. Muted?
No, it seems okay. So my question is, when you, for instance, apply your technique to study surface of a cell, not, for example, a protein embedded into a liposome, for instance, we have just a single class of a protein, but when you are looking at a real cell surface, and can you predict somehow which protein you're looking at the moment? For instance, you have like in the close vicinity several different proteins of different mass, of different everything. And is it possible somehow to distinguish between, for example, are you looking at, say, scramblase or, I don't know, ion channel or something else?
The type of protein versus, I mean, most of our distinction has been based on mass. So certainly we have that handle. There are other like I showed with the streptavidin, there's like some more complicated dynamics there. And then if you do the ear, then you get the full normal mode, which is a, sort of a fingerprint of the type of protein that you have. So depending on how much effort you're prepared to put into the experiment, you can get more and more information. We've also done just regular Raman on nanoparticles that are trapped. So potentially you could do just regular run-of-the-mill Raman on the particles when they're trapped there. And it would be interesting. I know that you guys have interest in just electromagnetic absorption, like direct IR, what would be termed IR type of absorption. So you can imagine microwave absorption type of experience. We've never tried that, but that is certainly would be a great thing to try to probe what you have there if you could do something similar with a microwave experiment.
Yeah, that's definitely the something I'll test out. Just the problem with the direct absorption is the water is still something which is covering the response of from the protein on the.
Yeah. So I did mention Shanghua is our collaborator. He does these photoconductive terahertz, and so we are working towards those types of experiments, but it is challenging.
Oh yeah.
It is challenging. Yeah.
There are probably some signatures of the like effective vibrational density of modes which can be probed there. But that's something which brings me to another question: is that when I was looking on the graphs you showed from here, what is the highest frequency of if you try to see the higher modes, because you mostly see only the first fundamental, but you try to go like higher, like if you sweep. So what is the practical limit for that, or you could see something interesting, or just not any particle peaks anymore?
I think we kind of limited protein studies to about 200 gigahertz or so. And that was at that time we were using 2 DFB lasers, so they're temperature tuned, and there's kind of a limit to how far you can tune them.
Okay.
But we did push further with an external cavity laser in this. I mentioned that we did some non-trapping experiments where we did nearly degenerate full-wave mixing type of experiments, where we basically cross 2 beams in a solution. And then it's not a single molecule experiment, but we looked at just gold nanoparticle seeds that were grown in solution, and the really tiny ones have like terahertz vibration. So we did push out to terahertz.
Okay.
So that was a nice experiment, being able to see a signature of those vibrational resonances in the terahertz regime.
Okay, and in bulk, basically, you say, right? So
that was in bulk, yeah. So that was not a single molecule experiment, and probably we should push out there. But we also had this discussion. I think people who do the normal mode analysis, most of these global modes, which would respond most strongly to our ear system, the ones that have the biggest sort of changes, have lower frequencies. They typically are in the range of, I would like to say, 20 gigahertz to just over 100 gigahertz. And I think it's energetically favorable from nature's point of view to work in that range. We discussed this previously. If you go below 10 gigahertz, say, then water can respond and really damp things out. If you stay above that 10 gigahertz range in the 20 gigahertz or so, then water cannot damp you out as much, I believe. But if you get too high a frequency, then there's too much energy per mode, and then so it's interesting that proteins are 30 kilodaltons or so, and that it happens to correspond to that size range. It's kind of a nice, maybe it's not a coincidence.
Yeah, that's very interesting. We discussed last week from perspective of how to nature develop or evolve the proteins to be basically using the physics of the surrounding media, right? Of the basically
exactly
of the water.
Yeah.
Interesting. Or kind of tuned to that to be
especially proteins that exist in solution, and membranes ones will be slightly different.
That's a good point. That's a good point.
Yeah, yeah.
So in short, for going higher modes, you didn't really go higher than 200, and about, let's say, this 150 or something. Could you see some more peaks? Because I don't think if you ever show the graphs of going like about that, like
We did
150.
have one. There was one really tiny, I think it was aprotinin or one of those that
Okay, the one in the
was up. No, that's streptavidin. Yeah, so there's a couple that are above 100 here, but streptavidin and the trypsin inhibitor, so it's some sort of serpine, I guess. I'm not sure if that is aprotinin or not, but certainly in the 100 over 100 gigahertz. But yeah, we haven't probed. You know, Turton's experiment where he's doing optical Kerr effect on lysozyme sort of proposed that those underdamped motions. I believe that was in the terahertz, closer to terahertz range. But if you look at his data, actually, there is some interesting stuff happening that he kind of ignored in the 100 gigahertz kind of and lower.
So did you check lysozyme just out of curiosity?
No, I don't think we did. Yeah, it's very common to do that. Probably we trapped it in egg white experiment, but we didn't actually do the. This is a common one for sure.
Okay.
Because it's so small and easy to model.
Yeah. So that's it. It would be easy to model exactly, but maybe the frequencies will be maybe about the what you typically see, like about maybe a few hundreds, because it should be corresponding to maybe a few hundreds of gigahertz.
Yeah.
Okay. Yeah, but I was asking about the higher frequencies because then the density of the mode should be higher. Like then there should be start to be some overlapping or some different behavior I would expect, because the first few with this line width, you can kind of isolate, but if you go higher, then the modes, if the line width would be the same, then the modes should start to overlap, and you would not probably directly see them. Just saying, right? It would be something interesting to see how does it connect to the this conceptual concept of the vibrational density of modes.
Yeah, no, this is true. Certainly, there's this sort of peak in the but I guess a lot of the questions about what is the biophysical relevance, a lot of that focus on some of the lower modes and them having the most importance in terms of large scale conformational changes and things like that. So that's kind of where we focus our attention. But certainly, there are more modes. I mean, the density of states goes up, and there's a kind of a peak there, right?
Yeah.
Yeah.
Okay. Cool. Okay. So, any more questions from any of our attendees? So it was now. Okay. So I think with this, I guess it's all. So I'll thank to all the attendees, our speaker, and I will stop the recording, and we can do whatever we like discuss of the record. So I'm stopping the recording now.
Yeah. Thank you.